publish all of these different papers into a focus issue dedicated to reporting and reproducibility in microscopy. Next slide, please. And really, this is, I want to emphasize again, this is really a global network of initiatives. Some of them have been around, like the Open Microscopy Environment, for a long time, and other ones like, for example, African BioImaging Consortium is a newcomer, but there is new ones coming pretty much every month, and we are all working together. And QUAREP-LiMi and BINA really working together with a lot of other organizations to achieve the goal of really improving all of these aspects, the quality control and reproducibility, and the ability to share. So in this presentation that are going to follow, you're going to hear about 9 different papers that were published in the focus. By the way, I wanted to highlight the cover, which was made by Tao Do at the Allen Institute, and it's really an amazing way of representing how metadata is really an integral part of the quality of the image. So actually the slides got inverted. So can you go to the next one? So in this, so the 9 different papers, there is going to be a paper by Global Bioimaging presented by Shuichi Onami about all of these different global initiatives to achieve common standards. And then there is going to be presentation on QUAREP-LiMi as a whole, on best practices and tools, on what is the problem and why we need to solve it. We will have a presentation about what EMBL-EBI is doing to solve this problem, and how we can develop new containers. The Open Microscopy Environment is gonna talk about, and Josh Moore is gonna talk about how we can develop new containers that are cloud ready for collecting all of this information. So go back to the previous slide now. And then you're gonna hear about some more from me about the community-driven metadata standards specifications that we have developed, and 3 tools. And you will hear from the authors about the 3 tools that were developed to be interoperable and to be able to help you collect all of this information. So in the first speaker is Shuichi Onami from RIKEN in Japan, and he will tell you about the global bioimaging collaboration to work together to improve all of this data management and quality control for image data. Shuichi?
Hi, so thanks for the introduction, Caterina. So my name is Shuichi Onami. I'm from RIKEN, Japan. So I'm talking about this paper, a global view of the standard for open image data format and repository. So this is a proposal from the Global Bioimaging Image Data Management Working Group. So I would like to first introduce the global bioimaging. So please, the next slide, please. Yeah. So the background of the global bioimaging is, as you know, that's in the biomedical imaging field, new technologies are rising. So including the light sheet microscope and the super resolution or others. So, and these advanced imaging technologies are often determine the quality or power of the research. So click, please. Click, please. So so these kind of the new images are made usually and operated by the skilled individual engineers by hand, and these technologies are always expensive. So when commercialized, so because of that, the problem is that many researchers cannot access to this kind of the advanced imaging technologies. So, and the solution should be open access to the advanced this technology, and also open access to the imaging data taken by this advanced technology. So, and this background, so we organized the global bioimaging communities. So next slide, please. So this is a global bioimaging. So this is an international collaborative organization that address the issue relating to the open access of the imaging technology and the data. So, and it is started from the Euro BioImaging, that kind of the similar organization, so specific for the European region. And so established in 2015, and it's a grow to the community around the world by bottom-up approaches. So, and now it is funded by the Chan Zuckerberg initiative. So, and so it is global bioimaging. So we have several activities and many activities that are listed here. So we have the annual meeting called the Exchange of the Experience. So we share the knowledge and experience of each country and each region. So, and also organize the thematic meeting and the working group to discuss about the specific problem or specific region of the problem relating to the open access of the data and the instrument. And also organize the training for these infrastructure people, and currently, so we are making more on the online training system. So, and also we implementing the staff shadowing program, so to share and disseminate these activities. So next slide, please. So, and in this global bioimaging, so we have the image data management working group. So, and so myself and Jason Swedel has been the co-chair of this group. And so from 2021, so that Josh Moore taken over Jason's role. So, and now we have that Jason and myself is leading this working group. So 17 members are and from the 11 countries are included in these communities, and we are discussing about the standardization of the data format or data repository was this kind of related the problem or issues. So, and through the discussion of this working group, so we made a proposal for the image data format and image data repositories. So next slide, please. The recommendation proposal of the data format. So we have, I don't want to go into it, but the data format standard should be have those 7. So the features, the openness, implementation, the example, licensing, the data type, and these the features should be implemented in the data standard, and the data standard important for the sharing the data and making the tool. So next slide, please. So we also make a recommendation for the image data repository. So we have 2 layers of the repository. So we call it repository for all data for all publication and added by the database. The repository should be that have the minimum metadata to be included in all data, but the added value database should have more richer metadata for use for high usability. So I don't want to detail, but we have several important aspects of the feature of this kind of the repository. So we already set this kind of the standard for the data sharing ecosystem. So, and we continue to moving forward to making this system working. So, and we want to collaborate with your organization to make this ecosystem working so worldwide. So, thank you very much for your answer.
Thank you, Shuichi. And next, we're going to have Matthew Hartley from the BioImage Archive. Talking about the BioImage Archive, is from the MBL-EBI, European Bioinformatics Institute, and BioImage Archive is one of these repositories that started implementing along the lines of global bioimaging recommendations. So thank you, Matthew, for participating. And
thanks so much for inviting me. So as Caterina said, I'm the team lead for the BioImage Archive, at the EBI, and we are in the taxonomy Shuichi introduced. We're a kind of primary archive for image data associated with publications. So we'll take image data from any bioimaging modality so that you can publish it as open access. And in order to make an archive work, next slide, please, we need metadata. We need metadata because without metadata, you can imagine the archive looking a little bit like the top left picture. It's a collection of sort of unordered stuff. We might have lots of amazing images, but we won't be able to find them, we won't be able to organize them, we won't be able to categorize them. So we need metadata in order to be able to turn our archive into something that looks a little bit more like the bottom right picture. Next slide, please. So in order to develop a metadata model that is very broad, because one of the problems with trying to cover the whole of bioimaging is it's everything from light microscopy, electron microscopy, atomic force microscopy. We even have, for example, handheld camera images of insects. It needs to be quite high level to cover these different use cases, but it also needs to be deep in certain places. So EBI coordinated 3 working groups to look at 3 different examples of modalities, and those were light microscopy, electron microscopy, and correlative microscopy, to try and come up with some common features that should be used to describe really any bioimaging experiment. And this gave rise to this high-level REMBI framework, and REMBI has 8 different modules, and those are the things in different colors. And the modules are really ways to categorize different important parts of metadata describing an imaging experiment. And they go from the very general, so the study is really describing what the purpose of the experiment was, why imaging was being carried out, who was doing it, through to much more specific things about sample, about how images were acquired, about the exact details of the image data, and so on. And if you're interested in the details, so next slide, please. In the supplementary material to the paper is the most detail that REMBI provides, which is a table describing some of the things that will go into each of these modules, the types of data that they cover, and then possible sort of formats or ways to categorize that data. So these differ, for example, describing an organism, it makes sense to use a species taxonomy. Describing, say, extrinsic variables, it might make sense to use a table. But importantly, and I think importantly for this group, for some of these categories, like describing the image acquisition, instrument attributes, it makes much more sense to use a well-established community schema, which is why the efforts like the 4DN BINA model, which Caterina is going to talk about later, are so important to us, because we need to be able to fit those into our schema where they give the necessary level of detail. Next slide, please. So it's important to kind of recognize what REMBI is and isn't. And what it provides is a very high level way to categorize different types of metadata and say roughly for a bioimaging experiment what should be described, which is the top, the green part. It gives some examples of what the data models for each of those components might be, but it's not very strongly prescriptive. And what it doesn't is provide a ready-to-go implementation. So there's no code associated with the paper. And that means that what we're doing at the moment is implementing an example version of REMBI for the bioimage archive, so that firstly we can use it to capture the metadata we need as part of our deposition system, but partly then we can show it as an example to the community of what REMBI would look like in practice, and then we can update it based on feedback from the community, particularly on sort of different subcomponents of REMBI and what's necessary, and then start to look at how it might benefit other groups that are doing image data management, because the more we can standardize at a high level across different imaging data management systems and repository, the easier it is to share data and let people sort of look consistently across the repositories. And we're always very keen to get feedback on what we're doing. So we've got an email address, or you're very welcome to get in contact with me. And we can talk about this more in the discussion section. So thank you.
Thank you, Matthew. And leading on to that, Josh Moore from the University of Dundee, and one of the leading forces in the open microscopy environment community, is going to tell us about their effort to develop next generation file formats. which are some of the implementation level aspects of how do we collect all of this information at the level of the file, so that then maybe can be deposited in these the repositories. So please.
We'll keep our fingers crossed. Yeah, thanks, Caterina. So hi, everyone. Right, in the interest of time, I'm going to just take for granted that everyone has experienced personally the difficulty of working with file formats in bioimaging, right? So it's something we all suffer with. And what I'll be showing, as Caterina pointed out, is work from a number of people and groups across various continents and software projects and data formats and all of that using data from somewhere else that's been published in IDR. So this, the Lemmers et al. Paper. So, you know, trying to bring the community together on fixing this ultimate problem that we're all dealing with. You can go to the next slide, please, Vanessa. Thank you. So, okay, so this is from the poster from OME this year. Trying to get everything in one slide so we don't have to click a bunch. So, like I said, we're all used to the problems of file formats in our domain. Obviously, that's where bioformats comes in. So you may have used it without even knowing. So very briefly, bioformats is a library to help all of us get our data out of the proprietary file formats that we're dealing with. So this is this A download path, right? So the status quo is really that you download an image and you use bioformats on the fly to kind of look inside of it and figure out what's the data, what's the metadata, what can I do with this? What we are experiencing, though, quite directly with, with the image data resource and other repositories will be running into the same problem, is that this is becoming a bottleneck, both maintaining all these different file formats, but also just in terms of speed. Trying to get access to your data is becoming more and more of a problem. And the same thing happens, so in this next path, so this B, you can put your data in a server and you can let the server do the same thing for you. It will translate the data on the fly. But ultimately, you still have the same overhead. You still have the problem of spending cycles trying to figure out what your data is. So on the one hand, we want to figure out a way to make the data open. How is it that we get past this problem that we're experiencing as a community? Some of us, and certainly everyone running data at EBI, also has an additional problem, which is how do we put this data into the cloud? So none of the file formats that we're currently using were built for living in the cloud. And we can talk about why that is later. So what we've tried to do here is develop a format that's open and transparent, really, to the end user. So no more using something to try to figure out what's in the file, break it open, store it in the cloud, and have as many different applications access that as possible. So we can go to the next slide. The major aha moment for me was something like this, where you have an application that's written for the browser. This is from Harvard Medical School called VISAR. And you have our data sitting in the cloud at EBI, and there's no server here. So people are remotely accessing this data. So this is the 30 gigabyte file. It could be much, much larger. It could be hundreds of gigabytes. It could be terabytes. You've put your volume of data in the cloud in a form that anyone can start accessing it. So the next slide. So we've done the same thing for Fiji and for Nepari, and obviously more people from the community are welcome to get involved. We'd like to see as much support as possible for these open formats. Next slide. So the real core of the paper that's in the issue is really about the performance. So what's the motivation for doing this? And I won't go through this slide, happy to talk about it, but the take home is as you move things into the cloud, so as you try to start scaling up, you have new forms of storage, the user will start experiencing a slowdown. The slowdown is particularly bad for other formats. Here, HDF5 is at the bottom. It's kind of the worst of the ones we looked at. We compared several different file formats. And the trick to make that work is by having broken your file format open and to put it in many different pieces, you can work in parallel. So, for anyone who's developing against cloud platforms, you need to work in parallel and try to get as much throughput as possible to your data. So that's the reason for the format. Interestingly, though, and I'll stop here, is what this also provides us is an opportunity to store our metadata in another way. So, it's clear we need more performance file formats. We're all working together on that. But this does give us an opportunity to take work from 40N, BINA, everyone else here, REMBI, and find a way to represent all of these in an open format along with the open data that's sitting in the cloud. Thanks.
Thank you, Josh. And next we're gonna have Roland Nitschke, that is from the University of Freiburg, which is gonna tell us about community work to bring all of this together.
Okay, so I will tell you a little bit about QUAREP-LiMi. And QUAREP-LiMi stands for Quality Assessment and Reproducibility for Instruments and Images in Light Microscopy. And as you see, it's so long, so the abbreviation really helps us to pronounce it properly. Can you go to the next slide? So we came together as a crowd, and you will see the crowd is a little bit different compared to the other one, because we try to bring together different communities which are usually not always working together. So, and we published 2 white papers, a shorter one in Nature Methods and a longer version of it in Journal of Microscopy. And more or less it was the work of people working in the so-named work group 9 of QUAREP-LiMi and all the chairs and co-chairs in the working groups. So next slide, please. So QUAREP-LiMi is really horribly quickly growing. Sorry for the poor quality. I think I made a mistake when I copied this into the presentation. Yeah, so you see we are covering now really a large majority of, I would say, countries doing microscopy. We still have some white spots and we are working on improving in that. We have grown now to 384 people, and we are still growing fastly, and the number of countries is also still increasing. Next slide. So here you see the development over time, and I think we very often see when we go to the public and tell what we are doing, then we have a steep increase in members in these months. So this month, for example, we were busy at the UK BioImaging meeting and at the AIM workshop in Berkeley. And you immediately see that you have new people hearing about what we are doing. Next slide. So who is in the community? I said we are trying to merge together people. And the starting point was for us, we had the national communities like BioImaging UK, BINA, Netherlands BioImaging, German BioImaging, the French imaging community, but we have also other ones like DIN and ISO members, the NIST, the BAM. These are standardization organizations, and we work tightly together with Euro and Global Bioimaging and OME. So all people who are working in these sub-communities, they are also in QUAREP, and the big white collection of icons which you see down there, these are all companies. So we have now 40 companies who are working with almost 100 persons in QUAREP-LiMi. And this is the approach we had to go, because in the end we need acceptance, not only in the people doing research or handling microscopes, but also at the companies producing instruments, because we need the metadata, and the best is to have the metadata actually already included when you do the experiment or when you do the imaging. Next slide, please. So we are organized in 13 working groups. We just recently expanded by 2 working groups by splitting up one of the working groups in 2, and we have a new phototoxicity working group. And each group is headed usually by 2 persons or 3 persons. We see that the amount of work is increasing, so it's better to have it on more shoulders. And that's the reason why in the end you can have also 4 people heading a working group. It doesn't matter. Next slide. Here you see how many people are working in the working group. So at least each of the working groups has more than 40 to 50 people, but some of them, they are 80 up to 100. This means not that at each meeting you have this amount of persons actively working there, but more or less you have around 20 to 30 people who are really actively doing work, acquiring data, because the working groups 1 to 6, there we have to record data. So this is even more, I would say, demanding to the members than some of the other working groups. Next slide, please. So what are the tasks or the duties in the working groups? For sure, if you want to get data, you have to decide what you have to measure or what you want to control or what has to be evaluated. This is more for the working groups 10, 11, and 12. And you have to define this very carefully. And then you have to define how to measure. You need software tools. You have to agree on the metadata wording and the definition, which is a big topic because we have the working group 7 headed by Caterina, which is about metadata, but we in our community, we have also to agree on the metadata. And this is not so easy. So we learn the working groups have to intermingle in between and have to agree on the same wording. Otherwise, it will not work at all. And in the end, we will analyze the data. We will bring out protocols, and some of the working groups already started with that. And this will be public. We will test it, and we will do it in a very similar manner to what the ISO and DIN committees are doing.
Roland, I think you have.
Yeah, I'm soon finished. Next slide.
Oh, you're...
Yeah, that's over.
Okay, thank you, Roland. And that is really important work. I'm sorry I had to kind of.
No problem.
So next, we're going to hear from Paula Montero Llopis, sorry, from the Micron Center at Harvard Medical School. And she has been leading a group of amazing people that are listed here to actually, together with other people, determine what is the actual problem with reporting and why do we need to report information, and how does that help us? And also they also have started developing tools to help doing that. So please, Paula.
Thank you so much, Caterina. And as you just said, what we try to do in this group is to try to explain researchers why it's important to do methods reporting. I feel like every imaging scientist out there understands like why methods reporting is very important. Everyone like included in this group are educators. We run core facilities that are like really focused in supporting our users. And what we have noticed throughout the years is that the main reason like people like don't do good methods reporting in light microscopy is not because they are like lazy or they don't want to, but like in most cases they don't really understand what needs to be included, why is this important. And we feel like one of the main reasons for this is the lack or like an inconsistent training in light microscopy and how to set up experiments, analysis, and everything. Can you please? Okay. So this is a very great, thank you. Yeah, so as I was mentioning, like really the goals of this project was like to focus on educating researchers, right? So let's try to get to their level and like help them understand what are the important parameters that are going to impact imaging, right? So what we try to do is to provide easy to follow explanations that like the researchers are going to be able like to understand immediately. So this is like really intended, our audience is for researchers, grad students, postdocs, people who use microscopy in general, but they are not experts. So in the publication, we try to help people understand through specific examples of figures that as soon as you look at this figure, people can immediately understand like how like that specific parameter is going to impact reproducibility. We also try to provide as many resources as possible that address this immediate need, which is to improve education on methods reporting. As part of the publication, we do identify and describe critical metadata for the most commonly used modalities. And what we try to do in here is to provide 2 tiers or classifications, if you want to call them that. First, those parameters that are really going to impact reproducibility and will affect the conclusions of your experiment. And then those metadata or those parameters that are important, that is good practices to provide, but they don't necessarily affect reproducibility. So we described or provide comprehensive checklists that what we try to do with these ones is so that authors, reviewers, or editors can use throughout the peer review process. And obviously, trying to educate people and trying to provide resources is not just enough. If we want people to implement these checklists or guidelines, it's better if we can help or facilitate with a tool. And so Rebecca Sem, who was in the first slide, was the person who created this tool. The resources that we are including in this paper tend to be visual so that people can understand better. This is one of the resources. It's a poster that summarizes the guidelines that we are including. So this can be downloaded, and we are working right now with vendors to be able to have it available for people for free. Yeah, perfect. And as I was saying before, the figures that we are including in the paper are easy, striking figures that people, even non-experts, are able to immediately understand what's going on. Next, please. Obviously, providing guidelines can be difficult since there are many parameters that are going to impact your imaging and the results. As you can see in here, this is an example of one of them. We try to go through the light path. Anything that is in italics is good to have, but it will perhaps not be impacting your data. But also, in many cases, people are gonna be using different. They're gonna be doing different experiments. They're gonna perhaps someone is doing that time lapse. Other people are doing multicolor imaging with different positions, right? So what we want to do is to make this checklist a little bit more accessible, and that is why we developed BitCheck. So it's based on a Shiny app. So it's gonna dynamically generate a checklist for the microscopy methods, depending on what the experiment that you did is about. So the user is gonna have
Paula, can you please wrap up?
Yeah. So so people are gonna go through easy questions, and then it's gonna give examples on the metadata that you can download. And really, the driving principles for this tool was to be as user friendly as possible, accessible, flexible, and adaptable. And I mean, the next slide is just for. We are also improving MidCheck. Rebecca talked about this in the AIM conference, the last one. So the new features. Yeah, the new features is for microscopy course and non-expert labs to upload their metadata. We want to have a wizard that will guide people who really are not experts in microscopy to how to get the information. It will store the repositories. You will get custom examples, and it will integrate existing tools and databases to gather the microscopy information. And what I'm excited about this tool is that then we can very easily also implement recommendations that we develop in the working group 11. So that's it. Thank you.
Thank you, Paula. And people are really appreciating the checklist and the tools. So we're going to talk a little bit more now about common standards, but also tools that are like MICCheck being developed to work together to help bring all of these specification into life. So in the first, sorry, can you just go back a second? Yeah, so the first paper I'm going to tell you about is really an effort that started from the 4D nucleome community, in particular from the Grunwald lab, and involved the OME community and BINA and QUAREP to really devise, next slide, devise shared community-driven specification for what kind of information needs to be captured, both in material and methods, but also importantly in the metadata associated with images to capture the image acquisition aspect of the metadata that is required to document an imaging experiment. So an imaging experiment has multiple phases: sample preparation, image acquisition, image analysis. The microscopy metadata specification really concentrates on the image acquisition. And as Matthew was saying, these are actually compatible with providing information, for example, for depositing on BioImage archive. So in the microscopy metadata standards specifications that we're developing are described two aspects. One is what is the provenance, where was the imaging coming from? So the specification for the hardware of the microscope, the image acquisition settings, the structure of the image is we define as provenance, origin of the data, and the quality control is everything that has to do with assessing the quality of the image and of the instrument. So these this specification really define what kind of metadata has to be captured to ensure the reproducibility, the quality, and the sharing value for image data. Next slide, please. So these specifications, I won't show you all the technical aspects of it, but it's really basically organized in a model, in a metadata model, which is a schematic representation of the hardware and the settings that can be utilized to acquire an image, and it's organized in a manner that is really schematic, such as, for example, in this case, we have an instrument that has a detector, and then we have information about each of these components. For example, information about the detector, information about the objective. So this was, we didn't want to start from scratch. So we started from the open microscopy environment model, and with 4DN and BINA and now QUAREP, we are working toward extending these specifications. And in order to make them as flexible as possible, next slide, please. Next. We are working on three different flexibility axes. One is we define what kind of experiments one need to perform and what kind of requirement a specific experiment might have. So an experiment doesn't require too much quantitative analysis, might require fewer metadata than an experiment that requires advanced analysis. We also have different extensions depending on the modality, and also we have different requirements for each field depending on the purpose. Next slide, please. But alone, these specifications are not able to help people. We need specific tools to allow people to work with them and bring them to life. So we have developed, so how do we capture all this metadata? So next slide, please. So in order to facilitate the acquisition of this metadata, you will hear about three tools: Micrometa app that I will tell you about, Methods J2 that Joe Ryan will tell you about, and Susanne Kunis and Stephanie Whitner-Kamps will tell you about OME-NGFF. And all of these tools are designed really to work together to bring to life the specification. So in Micrometa app, which was also developed together with the CIC, the Data Coordination and Integration Center of the 4DN community. Next, please. Is really a tool that allows you to collect the information about initially the hardware of your microscope. So it really brings to life a virtual representation of your microscope in a very intuitive manner to produce a JSON file that contains the information specified by the community specification. Next, please. And basically, so it's allows you to drag and drop different components, like a little bit of a video game, and allows you to enter information about each of the components. Next. The second part is it utilizes the description of the microscope and allows you to then extract information from your images to enter information about how the specific image or image data set was acquired. Next, please. And in order to do that, it utilizes bioformats to be able to, there is a missing slide arrow here, to actually integrate the information that is already present in the header of the file, and then allow you to integrate it with additional information to build the, build the light path for each channel and record all the information that you require. Okay, next, please. So this tool was already tested at 16 different facilities, and we have sample metadata files available on Zenodo for you to take a look at and play around with and see how they can help document different types of experiments and different experiment modality. Next, and then the next phase is going to be the integration of Micrometa app into Omero, so that it will allow you to integrate the metadata that is acquired using Micrometa app with the images you have in case you're using Omero. Okay, next is going to be Joel, who is going to tell you about another interoperable tool that is called MethodJ2 that works as a plugin of ImageJ.
Great. Thanks, Caterina. Yeah, so I'll be talking about MethodJ2, which we put together with this nice group of people here. So a lot of guidance to get this going. It essentially relies on the work that Caterina just presented, so Micrometa app, and we're trying to do this such that it follows the guidelines that were kind of established in Paula's work. So the sort of the guidelines to report microscopy methods. So if we can go to the next slide, please. Yes, so method sections for microscopy experiments tend to be incomplete very often when you go and read a paper. So you'll see something along the lines of images were acquired on a microscope and just perhaps say Zeiss. And the problem with this naturally is, of course, well, it makes it difficult to evaluate the work when you're looking at the images and trying to determine how it was done. And of course, this leads to poor reproducibility, when you're trying to replicate the work. And so our solution, one of the many solutions that were developed to this was to try to develop a text generation tool for imaging methods. I'll sort of walk you through this in the next couple of slides. So we put together MethodsJ2, which, as you can guess, is based on MethodsJ1. And so this is written as a script for Fiji. So we decided to use Fiji, of course, to leverage all the tools available in Fiji, especially bioformats, which allows us to go get the image metadata. And how it works essentially is that if we start from the left, the script will take in metadata from an image, so as best as it can source from it, as well as information from a Micrometa app microscope file. So this requires the core facility or the microscope custodian to build a Micrometa app that really details all the components of the microscope. So then this metadata, this information is taken in on the left. It's run through MethodsJ2 which is this Python script that runs in Fiji. And then it will prompt the user for a little bit of information and to make sure to either to specify or validate the information through a series of dialog boxes based on the image data and based on the microscope hardware, and will hopefully end up with a little bit of a blurb that can be copy pasted directly into a manuscript. And so that would be something along the lines of, images were acquired on a Zeiss wide field, and with all the information that can be in there, and hopefully, as much as we can, try to follow the guidelines as established in Paula's paper and as discussed by Paula just a little bit earlier today. So that's the kind of the main of it. The script essentially emits a little bit of a little text blurb that is copy pastable, and also not shown here, also a CSV file with all the input information that was either found by the script itself in the metadata or entered by the user. So also for documentation, you not only get that little bit of text, but an actual CSV file that you can use for yeah, for later documentation. And so we'll stop there. Essentially, the future directions of this are essentially, well, as you can imagine, we want to broaden the functionality. So at the moment, it works reasonably well with relatively simple images, so wide field, very simple confocal. But of course, we're hoping to bring in more of the hardware that is available to be selected in Micrometa app. So things like spectral detectors and more advanced functionality. We're looking for, of course, always community input. So I presented this and did a little demo at the AIM meeting last week and got a little bit of input from people and trying to do things like customizing and adding more information about acknowledgements and things like that. And we recently were awarded funding to rewrite this script itself to also run in Napari. So I have a little bit of a preview or a vision board, if you will, of what it would look like in Napari, so just this other editor that runs in Python. Currently, it's available on GitHub, and yeah, I'll stop there and happy to chat about some more at another time. Thank you.
Thank you so much, Joel. And the last but definitely not least is the work from Susanne Kunis and Stephanie Whitcomb-Peters about actually the grandfather or grandmother of all of these tools, which is OME-NGFF and its current implementation, and MDEmic, which is the current version of it.
Yeah, thank you, Caterina.
Susanne.
Yes, my name is Susanne Kunis. I'm working at the Cernannos and at the University of Osnabrück. This is a multidisciplinary center with light and electron microscopy, and I want today to report on MDEmic. This is a metadata. Okay, this was a little bit too quick. It's okay. A metadata annotation tool to facilitate the capturing of microscope relevant metadata. Next slide, please. So what is our starting point? We want to make imaging data fair, and a significant factor of this are the metadatas. Collecting and maintaining the relevant metadata is the key to ensure that the data are later reliable and reusable. And the individual researcher in the lab should be able to, to add the complex metadata of a microscopy experiment in a comfortable way. The annotation with the metadata should take place in very early research process when all information is available. So you see here the researcher in the lab, and he has made his acquisition and want to upload now to a data management system. In our case, it's Omero. And this is a good starting point to add metadata that are relevant to this microscopy experiment. Next slide, please. Yes, a little bit to the user interface. For this process, we developed, for this metadata annotation process, we developed the MDEmic to support the user in this capturing process of metadata and to enable the integration of recommendation metadata standards, for example. It is composed of different components with different level of user support. At the basic level, we have structure and vocabulary of the OME schema that are used as to provide the user as input forms. And then on the second level, bioformats is used to read out all the metadata stored in the image file to this OME schema and load these values into the input forms. But bioformats not supporting all file formats so well, and to give the user support and annotate also this data, we have the third level where we could customize in one way the metadata, the used metadata schema, and to provide a standardized vocabulary and structure to annotate the data. And the last level is you could also integrate pre-definition of individual object. That could be hardware or instrument description that the user can use to annotate the data. So where bioformats fails to read out the metadata from the file, the user can add it with one click. Next slide, please. We have integrated the MDEmic as OME-MDE into the OME importer. This is a tool with a graphical user interface for transferring image data to the OME repository. So the tool is at the moment part of the standard OME Insight installation and can be used within this tool. The components like the metadata structure and the metadata input forms of the OME-MDE can, as I mentioned, customize by the means of a configuration file. And OME-MDE stores then the collected metadata as key value pairs in the OME database with associated with the images in the repository. In addition, the metadata could be exported directly from the MDE in other file formats like CSV, TSV, and text format, and also in an ISA tab format that also IDR use for their metadata annotation. Next slide, please. As a use case with a direct benefit, we show in the publication an example from the University of Düsseldorf, where image data from samples treated with various membrane dyes and should be made available in a membrane dye database to the user. This database is hosted in institutional OME instance and shared between the members of a collaborative research center. And in OME-MDE, we have integrate object for the detailed description of the membrane dyes and for the technical description. And the user can use this to annotate the data and enter the data in a standardized way during the transfer step to the database. And these achieve a uniform metadata annotation and thus comparable and searchable data in this database. Thank you.